Thank you to the organizing committee for this opportunity to provide our presentation titled Analysis of Molecular Mechanisms Associated with Low Temperature Induced Freezing Tolerance and Floral Competence in Camelina sativa. Today, I will provide an overview of the objectives, describe the experimental design used to phenotype for cold-induced freezing tolerance, describe the differential abundance of transcripts observed between a spring and a winter biotype of Camellina in response to a cold acclimation time course, provide an overview of gene ontologies enriched with transcripts in a winter biotype of Camellina, discuss transcription factors that likely regulate transcript expression in Camellina, and provide conclusions. Camellina is an old world oilseed crop of the Brassicaceae family that is valued as a source of seed oil for its industrial applications and its ability to grow on marginal landscapes. Camellina consists of both spring and winter biotypes. As observed in this slide, spring biotypes, such as Co46, generally flower without a cold treatment, whereas winter biotypes, such as Joel, generally require a cold treatment to flower. As observed in the lower picture, cold acclimation of winter biotypes also induces freezing tolerance, whereas cold acclimation of spring biotypes does not induce the same level of freezing tolerance. Two hypotheses have been proposed for the heritability of low temperature induced freezing tolerance in Camellina. One proposed by Speroni et al. 2021 suggests freezing tolerance involves additive effects of genes, whereas another proposed by Horvath et al. 2019 suggests that freezing tolerance involves several dominant upstream regulators. Because the work by Horvath et al. Was only conducted on plants that had been exposed to 0 and 8 weeks cold acclimation, in this presentation, we explored differential expression of transcripts at multiple time points during cold acclimation of the same spring and winter biotype. Our experimental design starts with exposing 3-week-old Camellina seedlings to cold acclimating conditions at 5C under a 12-hour light-dark cycle for 0, 1, 2, 4, 6, and 8 weeks prior to subjecting them to an 8-hour ramp down in temperature to -15°C. After a -15°C freezing treatment for 4 hours, the plants are ramped back up to 20°C over 8 hours and then moved to the greenhouse for phenotyping for survival over a 42-day period. The slide to the left shows the growth and development of the winter biotype of Camellina following 3 weeks at ambient greenhouse temperatures following the freezing treatment. The slide on the right shows the visual survival rating of plants over a 42-day period in the greenhouse. Note that freezing survival was dependent on the duration of cold treatment, and plants that received less than 2 weeks cold did not survive the freezing treatment. However, all plants that did survive the freezing treatment also flowered, but initiation of bolting and flowering was dependent on the cold treatment duration, with plants receiving 6 weeks of cold bolting and flowering first, followed in order by plants exposed to 4 and 2 weeks of cold. Overall, after 42 days in the greenhouse, winter biotype plants receiving 2 weeks cold had 20% freezing tolerance, with plants receiving 4 and 6 weeks cold showing 32 and 72% freezing survival. Our earlier studies confirmed that 8 weeks cold consistently results in 100% freezing survival of the winter biotype Joel. Thus, we did not include an 8-week treatment for phenotyping freezing survival in this follow-up study. At each cold acclimation time point, we collected leaf tissue from both the spring and the winter biotype for conducting RNA-seq analysis. We used a very stringent approach of FPKM greater than 2 and FDR of Q less than 0.05 to identify transcripts showing differential levels of expression between the spring and winter biotype at each cold acclimation time point. Of the 89,418 Camellina genes annotated to Arabidopsis, we identified between 2,083 and 6,347 non-transcription factors, which are shown in the upper graph, and between 139 and 522 transcription factors, shown in the lower graph, that were differentially expressed between the spring and the winter biotype in response to cold acclimation, which are represented by the blue bars in each graph. After removing transcripts showing duplicate annotations to adapt Arabidopsis genes, which are represented by the orange bars, we sorted transcripts by those with log2 values greater than or less than 2, which represents a 4-fold increase or decrease in expression in the winter biotype compared with the spring biotype, and used that set of transcripts to conduct gene set enrichment analyses. Gene set enrichment analysis helped to identify gene ontologies enriched with transcripts with increased, represented by the blue text, decreased, represented by the red text, or both increased and decreased, represented by the green text, expression in winter biotype Joel during cold acclimation. Due to time constraints, I will only provide an overview here. Quickly looking across the gene ontologies for biological process in the top portion of the figure, you will notice that abiotic and biotic defense responses are observed at all cold acclimation time points, which included responses to cold, dehydration, heat, salt, wounding, bacteria, fungus, herbivores, and insects. Early responses to cold seem to be associated with ABA and jasmonic acid signaling processes, whereas ABA, auxin, cytokinin, and salicylic acid responses are more prominent with the duration of cold acclimation. The main processes impacted by cold acclimation appear to involve oxidative stress, transport of lipids, ions, and water, carbohydrate metabolism, photosynthesis, flavonoid and anthocyanin biosynthesis, and protein biogenesis and targeting to membranes. Based on the GO for cellular component across the bottom of the figure, it appears that these processes impact extracellular regions, cell walls, apoplast, chloroplast, vacuoles, membranes, and plasmodesmata. Among the differentially expressed transcripts annotated to Arabidopsis transcription factors, many shown in the far right column under process are associated with cell progression and expansion, flavonoid and anthocyanins, flowering, growth regulation, light signaling, and of course, stress. I will only discuss the potential role of several of these transcription factors may be playing in cold acclimation and cold-induced freezing tolerance in Camellina. MYB75, 90, and 111, and HAT1 are all involved in anthocyanin production, and their increased expression in the winter biotype may contribute to antioxidant defense processes. Growth regulatory factors are involved in regulating growth inhibitors such as DELAs, including RGL1. Thus, the increased expression of GRF7 would fit well with the observed decrease in RGL1 during cold acclimation. And the increased expression of PIF3-like would also fit well with the known repression of PIF genes by DELAs. Of course, many of the defense responses observed during cold acclimation are likely to be part of the CBF regulon, and CBFs do show a trend of increased expression in the winter biotype. Of particular interest are two nuclear factor Y genes, NFYA4 and NFYB2. These two subunits of NFY complexes are inversely expressed in the winter biotype at all cold acclimation time points. In Arabidopsis, NFY subunits are known to form heterotrimeric complexes that bind to target gene promoter elements to regulate their transcription. Also of interest is an MYB47, which is cold-induced in the winter biotype of Camellina, but was essentially shut off in the spring biotype. Based on Arabidopsis experiments and highlighted by the red arrows to the left figure, MYB47 is induced by methyl jasmonate, oxidative stress, wounding, drought, salt, and cold. In the right figure, the red arrows show the promoter of Arabidopsis MYB47, which contains a high 5 and a HAT1 motif that are likely involved in regulating MYB47. Although high 5 was not differentially expressed between the spring and the winter biotype of Camellina, it was cold-induced in both biotypes. In Arabidopsis, cold is known to induce HY5, which is a central regulator of photomorphogenesis and is known to induce expression of MYB transcription factors and to regulate processes such as cell elongation, cell proliferation, chloroplast development, pigment accumulation, and nutrient assimilation, which fit well with the cold-induced processes observed in this study. Thus, the putative MYB47 transcription factor in Camellina could be playing an important role in cold-induced freezing tolerance. In conclusion, freezing tolerance in Camellina appears to involve additive effects of genes associated with cellular components, including cell walls, extracellular regions and apoplasts, vacuoles, chloroplasts, plasmodesmata, and membranes. However, our results also suggest a small number of upstream transcription factors, including MYB47, MYB75, and MYB90, NFYA4 and NFYB2, HAT1, GRF7, PIF3-like, and CBF1 and 2 likely regulate transcripts involved in abiotic and biotic defense responses, anthocyanins, cell expansion, photosynthesis, transport of molecules, and phytohormone signaling. The observation that transcripts annotated to NFYA4 and NFYB2 show an inverse relationship in expression in response to cold acclimation is noteworthy. Thus, because these two subunits of NFY complex are differentially regulated by cold acclimation in the winter biotype, it is possible that these two transcription factors constitute a critical temperature sensing switch for regulating networks involved in cold-induced freezing tolerance. In closing, I would like to acknowledge my collaborators on this project, Dr. David Horvath and Dr. Winshaw, and my support scientist, Brent Bigger, who are all of the USDA ARS, and Dr. Marisol Birdie and her graduate student, Michaela Neubauer from North Dakota State University. Thank you.